So today is 10/3/17. I would strongly recommend that while we're taking notes, which is what we're going to be doing, you also have your vocab out near you. We've hit a lot of these words. We're going to pick up a few of the last ones today. Your quiz on this stuff in here is going to be Friday. So we're going to get through some material, and then I'm going to hand you study guides, and you can start picking away at those, and those will be due Thursday. Okay, so that's sort of where we're headed for the week. Now, why do we care about building proteins? Because we want to think about how bacteria can end up surviving an antibiotic. Bacteria have a very special protein coating on the outside of them, and a lot of antibiotics actually attack the process that bacteria use to build proteins. Now, we're starting off not with bacteria, but with eukaryotes. But the processes are similar. We're going to start to compare and contrast those tomorrow or Thursday. What is it called when we build something? Producing. What else? Constructing. All the synonyms you can think of for building something. Making. You've heard of this thing called photosynthesis that plants do? You synthesize things, you put things together from raw materials. We call the process of building proteins, and I realize we haven't really said this before. Protein synthesis. Anytime we use the word synthesis, we're talking about putting something together from smaller units or from raw materials. So you could go home and synthesize a batch of chocolate chip cookies. Actually, that sounds pretty good. Okay, so we're going to build a protein. That's what we're doing. Where do we start? Where do we start? Where do we start? Where do we start? This is review. This is okay. We're backing up to make sure you got the basics. We start with DNA. Yeah. The blueprint. The blueprint for building proteins in your cells, a maple tree's cells, a blue whale cells, a mouse, an earthworm, a ladybug, a firefly, a bacteria, this plant. Same thing. They use DNA, and to make a protein from a section of DNA from a particular gene, because remember just a portion of DNA is called a gene, we have to unzip a little bit of it. So I've already shown it unzipped. So we've got a sequence there, but we have a problem. What's the problem? Can't get out of the nucleus. Too big, too bulky, too messy, little tiny nuclear pores. We can't get the DNA out of the nucleus. We have to make a smaller copy. We have to copy it over onto some smaller, more convenient form. What's that smaller, more convenient form? Talk about it at your table. What's that smaller, more convenient form? I'm spying on you all. Hearing some of the right answers. What's that smaller, more convenient form? A few people said RNA. Hi, Tiffany. A few people said RNA, yes? And it's specifically messenger RNA because it runs the message out to where the proteins are going to be built. Okay, the process, when we go from DNA to RNA, we're copying it down. Are we changing the language? No. It's a different accent, but they're both nucleic acids. These are both nucleic acids. So we're not changing the language. We're just transcribing it. We are going to transcribe it into mRNA. So let's do this one together. What does a T complement? T in DNA complements what in mRNA? A. Yeah. Awesome. Now here's the tricky one. What does an A? U. Okay. What does a C in DNA complement in RNA? G. These are the easy ones. And G's complement C's, C, C. T complements, now we're down to repeats. A. A complements what? U. Messy U. And then the C complements a G, and the T complements NA. Okay, so we now have a strip of mRNA. We can take that puppy out of the nucleus, out through the nuclear pore. And what we really want to get is a protein. That's our end goal, right? We're building a protein. If we're you or me, we're building a protein to create the structure of our eye, to create a section of muscle, to create something that will allow our blood to clot. If we're a bacteria, we might be constructing a protein that's going to help protect us from harmful chemicals in our environment, like antibiotics. How do we get from mRNA to a protein? We started to talk about this yesterday. We did a little bit of practice, but I wanted to make sure we had the process, just the overview. So we take that mRNA, it goes out to the where? To the ribosome, out in the cytoplasm to the ribosomes. And at those ribosomes, little tiny delivery vans pull up. And those little tiny delivery vans, well, those little tiny delivery vans are the tRNA. So T for transport, T for truck, however you want to remember it. Those little tiny sections of nucleic acid, of RNA, pull up to the ribosome, and each of them are carrying a specific amino acid. We are actually changing the language now. We're no longer in the language of nucleic acids. We're now in the language of proteins. It's a chemical language, but it's a language. So when we go from one language to another, what do we have to do? Translate. This step is called translation. If you have ever translated anything into another language, how many of you are in a foreign language this year, in Spanish? Okay. So you know what it means to go from one language to another, to translate. And here we're translating from nucleic acid language to protein language. Chemical languages, but languages nonetheless. So that tRNA has a sort of a license plate. It's kind of a license plate. And that license plate just provides a check to see if it's the correct amino acid that's coming over. So, AUG matches UAC. Yep, check, that's the correct one. So now this tRNA can disappear. Goes back out into the cytoplasm to deliver another one. The next little van sort of pulls up. The license plate, does it match? CCA. Because remember, we're looking at segments of this at a time. What are those three base chunks in mRNA called? Codons. So, does the anticodon on the tRNA match the codon in the mRNA? CCA, GGU. Yeah, it matches. Okay, so we can pull this amino acid into place. The tRNA disappears. These things get linked together with a special kind of chemical bond. Do you remember what it is? It's a peppy one. Peptide. Peptide bond. So these are polypeptides because they're linked together with multiple peptide bonds. And then, we've just shown 3 codons here. So that last tRNA comes up, checks, UGA, ACU. Yep, that matches. You can pull this one into formation, link it together with the peptide bond. Terrible handwriting, I apologize. And the tRNA can go away. That's the overview. The process at the level that we're talking about it isn't all that complicated. So we transcribe from DNA to mRNA, and then we translate from RNA to proteins that make up all kinds of things. Now, what I'd like you to do, as you finish up your illustration, is get out, and some of you already have it out, that codon chart, because I want to look very specifically at a couple of codons. So we're looking at our circular codon chart, and notice that I didn't name those amino acids that we put up there. I just called them generic amino acids. I made them look like blobs of ice cream or I don't know what, just sort of pastel-colored blobs. But you know that every codon codes for a specific amino acid. So I want you to look at AUG. Go through the codon chart process. The codon chart, remember with the circular chart, you're starting in the inner ring. So we're starting with A as our first base. And then we're looking at U. And then we're going to G. So what is the amino acid that's being coded for? Go for it. Methionine is how it's usually pronounced. Methionine. Methionine is special because it starts every protein there is. Okay? Methionine, AUG, is also called the start codon. The way that a lot of people remember this is school starts in August. AUG is the start codon. If there is a start codon, a protein begins to be created. If there's not a start codon, you're not starting a protein. Now, if you know when to start something, what else should you know? When to stop it. So, if you look at, and let's check out this codon. UGA. So there's only one start codon. And let's see what UGA specifies. Let me clear that. So if I go to my first letter is U, my second base is G, and my third base is A, what is this codon code for?
Stop.
Stop! Stop making the protein. That's enough. Enough amino acids. We're done. So this is a stop codon. Now there was only one start codon, but I want you to count how many stop codons you can find on here. There are, well, it appears in two places, but remember that in some cases, more than one codon codes for the same thing. So UGA codes stop, UAG codes stop, and UAA codes stop. So there are three different ways that a codon can code for stop. We're done. We're done with the protein. Could you have a mutation that artificially shortened a protein if it changed an amino acid to a stop codon? Yeah. And that is something that happens in a few cases where there's a single base mutation. What do we call that? What kind of mutation? Point mutation. Yeah, look, right there. I'm pointing. Get it? Ha 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 ha. Yeah, we can have a point mutation that instead of coding for an amino acid, accidentally codes stop. That's a problem. You get a shorter protein than you would otherwise. So right now, what I'm going to do is give you some time. If you did not finish the chart on the front, I want you to work on the chart on the front of this. And then I want you to work on the back. And then we're going to talk a little bit more about how it is that some mutations have no effect. Okay? I'm going to talk a little bit about this. So if you had a section of DNA that was A. So the RNA complement for that would be what? U. Hey, codes for phenylalanine. Yes? What if I had a point mutation? And this A was mistakenly replaced with a G. What would be the complement? UUC. Would that cause a difference in the protein that was produced? No. Same amino acid. The fact that there are more possible combinations of mRNA bases than there are amino acids is one of the reasons we can sometimes have a mutation that doesn't actually have any effect, because it still produces the same overall protein because the amino acid is not changed by the mutation. That's an important point. We're going to look at some air test questions probably tomorrow, and I want you to keep that in mind. All right. The mRNA is the codon. tRNA is the anti-codon. And I realize that that's not showing up underneath the logo there. So tRNA are the anti-codons. So you have codon matching anti-codon. Okay, so we go from DNA to mRNA. The tRNA delivers the amino acids. They get hooked together with peptide bonds. We form a protein. That's pretty straightforward. Now, chemically, it's fairly complicated, but at the level that we're doing it, like I said, it's not all that complicated. Questions? Comments? Yes, ma'am. So those amino acids they're very often called building blocks of protein. They're small chemical units. And those small chemical units link together to form proteins. So if you think about, who here ever played with Legos as a kid? We're in the Lego phase of life at my house. We're actually still in Duplos, but I love them. Sometimes I even let my daughter play with them. Each piece is an amino acid. The structure that you build with them is the protein. So they literally are small chunks that can be put together to form a protein. Other questions? Comments? Concerns? Okay. So tomorrow, the plan is this. We're going to talk a little bit about comparing and contrasting protein, what do we call it when we build a protein? Protein synthesis in eukaryotes, like you and I and maple trees and blue whales and earthworms and ladybugs and lightning bugs and cockroaches and dandelions, and prokaryotes, which are basically bacteria and bacteria-like organisms. Okay? When you are done with that worksheet, you're going to put that in the box. I'm going to get you, actually I'm going to direct you up here. The study guide for this little chunk is right here, and you can start working on it. And those are going to be due Thursday. So tomorrow we start the compare and contrast. Friday you take your quiz, and you'll turn in the vocab then. How many of you are all done with Part C for the vocab? Okay. I think we've talked about pretty much every word at this point. If there are ones that you're still not sure of, discuss them with your friends. Well, I think this is this, but what do you think? Because that engages that social part of your brain and it lights up more neurons in between your ears, and lighting up neurons is good. We like lit up neurons, like little Christmas trees in your skull. Yay! That's it.